the membrane-bound organelles are all kind of hooked together by this system called the endomembrane system, or the membranes that are inside the cytoplasm. So the endomembrane system includes the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, lysosomes, vacuoles, and then the plasma membrane, which is surrounding the entire cytoplasm. So all of these components are either attached to each other or they can be connected to each other by transfer bubbles of membrane that are called vesicles. So a vesicle is basically like a little bubble of plasma membrane that might have stuff inside it. Think like a balloon. With the balloon, the walls of the balloon are made out of phospholipid bilayer. Okay, so endoplasmic reticulum. What a mouthful. What does it mean? It means inside the cytoplasm, and reticulum means little net. So it kind of looks like a net under the microscope, which is how it gets its name. And we're just going to call it ER from here on out because that's a lot easier to say. And the ER makes up an awful lot of the membrane in eukaryotic cells. And it is continuous, it's attached to the nuclear envelope. And then there are two flavors of ER. There's the smooth ER, which doesn't have ribosomes on it. And then the rough ER is so called because the surface of that ER has ribosomes all over it, attached to it, bound to it. So here's a picture showing, here's the nuclear envelope, which is shown in this sort of darker purple. And then it's shown as sort of morphing into the rough ER, which then morphs into the smooth ER. But it's really all part of the same thing. They aren't different colors in a living cell. They're just kind of drawn that way here to give you that impression that they are discrete, separate things. But the rough ER has all of these ribosomes attached to it. The smooth ER does not. And the functions of the smooth and the rough ER are a little bit different. The transitional ER is just, it's kind of looks like rough, but it's not really rough or smooth. It's kind of in between. So here's an electron micrograph, or basically a very zoomed in picture with a very fancy microscope showing you a section with rough ER with all these ribosomes attached to it, and then smooth ER with no ribosomes attached to it. So because the rough ER has ribosomes attached to it, those ribosomes, their job is to make protein. And the rough ER can take those proteins, and if they're going to be exported outside of the cell, they can prepare those proteins for shipping out. So transport vesicles are, imagine they're kind of like little bubbles of membrane that have these proteins in them, and then they're kind of getting their final gussying up before they get shipped out of the cell. So if a protein needs, like a heme group, like in hemoglobin, or the quaternary structure needs to be folded a certain way, that all happens in the rough ER. And it's also a place that produces new membrane for the different structures in the cell. The smooth ER that's the ER with no ribosomes on it. It produces lipids, so fats and things like that. It can also help to break down and build up carbohydrates, like glycogen. Also, it can help to detoxify drugs and poisons, which to a cell, not much difference between a drug and a poison, but it's a foreign compound to the cell. So the smooth ER can help us to metabolize, like if you're going to go out drinking, you hope you have a whole lot of smooth ER that can help you process it and not build up toxic byproducts of that. Another thing it does is it stores calcium ions, calcium 2 plus, which is very important for a number of cellular things. And your body needs calcium for muscle contraction. So that's an important function of that smooth ER. All right, next part of the endomembrane system is called the Golgi apparatus, sometimes called the Golgi body. And the function of the Golgi apparatus is shipping and receiving. It's kind of like the UPS store of the cell. So the Golgi apparatus can take those transport vesicles that come from the rough ER and export it out of the cell. So it can take some of those products from the ER and make sure that they are really good and set to go outside the cell. Also helps manufacture some types of large biological molecules. And sometimes it might put little tags on, molecular tags on different molecules to say, okay, this needs to go into the bloodstream, or this needs to go to the brain, or this needs to go to some other part of the body. The Golgi apparatus is kind of like putting a shipping label on those molecules so that those molecules know where to go. And here's a picture of Golgi himself. He was an Italian scientist, and he had a really awesome looking mustache. So how do you know the difference between the ER and the Golgi bodies? The Golgi apparatus typically looks like a stack of pancakes on top of something. So imagine that there's like a big blob of butter down here and you stacked pancakes on top of it. So it kind of looks like a series of letter C's on top of each other. And that actually tells you there's a directionality. So there's a cis face, which receives vesicles, things getting ready to be shipped out of the cell, and then a trans face, which is where the vesicles that have the finished products release those products. All right, next part of the endomembrane system are the lysosomes. And lysosomes are ways for cells to go through hydrolysis, which is that way to break down macromolecules by using water to add in and break off monomers of very large polymeric macromolecules. So those lysosomal enzymes can break down proteins, fats, polysaccharides, and nucleic acids. And inside those lysosomes, inside those compartments, one of the ways they are capable of doing that is that they have a low pH, meaning that they are acidic, and that helps to facilitate breaking down those large molecules. It's kind of like these are like the trash compactors of the cell. So sometimes cells need to have a very large bubble in them called a vacuole. Plant cells and fungal cells may have vacuoles, and these are basically like great big vesicles. So vesicles and vacuoles are essentially made out of the same stuff. It's just that vacuoles are very big and vesicles tend to be rather small. So animals can also have vacuoles. A lot of your single-celled eukaryotes they don't have mouths, so they will eat by producing food vacuoles for cellular eating called phagocytosis. Some other protists that live in water will have a contractile vacuole, which kind of helps to keep them from getting waterlogged, from like bursting because so much water is trying to sink them. So they can kind of bail out using these contractile vacuoles. And then in plants and fungi, they may have a large central vacuole, Organisms that have a cell wall need a central vacuole to make sure that the plasma membrane is always attached to the cell wall. Okay, I think in the next video I'm going to tell you about mitochondria and chloroplasts. These are a little bit of a different type of organelles, so they're going to get their own new video for that.